Deep within the cell's nucleus, there's our DNA. DNA is made up of genes, and each gene is basically a specific part of the DNA that codes for a protein. And genes become proteins in two steps: transcription and translation. Transcription is the first step in creating a protein, during which a specific gene is read and copied on an individual mRNA, or messenger RNA molecule, which is like a blueprint with instructions on what protein to build. Translation is the second step, and it's when organelles called ribosomes assemble the protein from amino acids lying around in the cytoplasm. Each mRNA has a direction, running from the 5' end toward the 3' end. mRNA is a chain of 4 types of nucleotides, which are the individual letters or building blocks of mRNA. Nucleotides are made out of a ribose, which is a 5-carbon sugar, a phosphate, and one of 4 nucleobases: guanine, uracil, adenine, and cytosine, or commonly GUAC for short. Guac. These nucleotides are the actual information carried within the RNA. Each 3 letters is 1 amino acid. In total, these 4 nucleotides can be arranged into 64 combinations. The freshly made mRNA floats out of the nucleus through a pore, and hooks up with an idle ribosome to start getting translated into a protein. Right away, there's a process called initiation, which is where the ribosome grabs the mRNA. Groups of 3 letters are called codons, and each codon codes for 1 amino acid, or gives a certain signal, like to start or stop. So for the ribosome to start, it needs to find a start codon. That's usually a sequence of AUG, which simultaneously codes for the amino acid methionine. So most proteins actually start with methionine. When the ribosome runs into this, it knows that from here on out, every subsequent codon represents 1 amino acid in the protein. Now, while each codon codes for a single amino acid, each amino acid can be coded for by 1 or more codons. For example, while only the UGG codon codes for the amino acid tryptophan, as many as 6 codons code for the amino acid leucine. UUA, UUG, CUU, CUC, CUA, and CUG. Translation continues until the ribosome runs into a stop codon, which is a sequence of UAA, UAG, or UGA. The stop codons don't code for anything else, except for stopping, which is also known as termination. But mRNA is only a blueprint of which amino acid goes where in order to get a protein. The actual job of ferrying the amino acids to the ribosome is done by another type of RNA, called transfer RNA, or tRNA. tRNA is a very short RNA sequence and is specialized for finding a specific type of amino acid within the cytosol and bringing it to the ribosome. tRNA only has a 3-letter coding sequence, called the anticodon, which is complementary for what's written in the mRNA. It means that it can briefly bind to the corresponding mRNA codon and deposit the amino acid within the ribosome. So the ribosome has 3 sites for tRNA to bind. They're the amino acyl site, or A, the peptidyl site, or P, and the exit site, or E. The A site binds the incoming tRNA with the complementary codon on the mRNA. The P site holds the tRNA with a growing polypeptide chain. And the E site holds the tRNA without its amino acid. So how this all works is tRNA, carrying an amino acid, initially binds to its corresponding codon on the mRNA at the A site. Then a peptide bond, which is the bond that holds the growing protein together, forms between the amino acid of the tRNA in the A site and the amino acid of the charged tRNA in the P site. The growing polypeptide chain is transferred to the tRNA in the A site. After that, everything moves by one site. The now empty tRNA in the P site moves to the E site. The tRNA with the polypeptide chain moves from the A site to the P site. The A site becomes open for a new tRNA to bring another amino acid. Finally, the tRNA in the E site leaves, grabs another amino acid in the cytosol, and returns to float around the A site to repeat the process when needed. So as the ribosome slides down the length of mRNA, it leaves behind a polypeptide tail, which is our growing protein. So if the mRNA says the following, 5' end AUGGUACCC UGA 3' end, that means that AUG is the start codon, and immediately it requires a complementary tRNA with sequence UAC, which carries a methionine. Next codon, GUA, requires the tRNA with the sequence CAU, which brings a valine. The next, CCC, needs an anticodon GGG, which carries glycine. Finally, there's UGA, which is the stop codon, and translation is done. Our tiny little protein is made out of just 3 amino acids, methionine, valine, and glycine. The final part of the process is the ribosome ferrying its freshly made protein to an organelle called the endoplasmic reticulum, where it can get further prepared for whichever role the cell might have for it. Alright, as a quick recap, translation is a process of reading the mRNA within a ribosome, and assembling a protein based on the instructions found within. mRNA is made out of codons, which are triplets of nucleotides that either provide start and stop information, or are coding for a certain amino acid in a protein sequence. The amino acids are supplied by the tRNA, which contains an anticodon, which is a complementary sequence to the mRNA codon. The translation is finished upon reaching the stop codon, which is the signal that the protein is done.